Good day. This is the Professor, CBMD PhD, and today is Wednesday, May 24th, 2023. It is 4:02 and 2 seconds, p.m. Eastern Daylight Time, and today's research, engineered bacteria. So let's hop to it here. Engineers create bacteria that can synthesize an unnatural amino acid. Date May 23rd, 2023. Source: University of Delaware. Researchers have engineered bacteria to synthesize an amino acid that contains a rare functional group, and others have shown to have implications in the regulation of our immune system. The researchers also taught a single bacterial strain to create the amino acid and place it at a specific site within target proteins. These findings provide a foundation for developing unique vaccines and immunotherapies in the future. The research laboratory uses tools from synthetic biology and genetic engineering to create microorganisms that can synthesize different types of compounds and molecules, especially ones with functional groups or properties that are not well represented in nature. The nitrochemical functional group has valuable properties and has been underexplored by folks who are trying to rewrite metabolism. As PNPAT also has a nice history in the literature that can be added onto a protein from a mouse delivered back to mice, and the immune system will no longer tolerate the original version of that protein. As that ability has promise for the treatment or prevention of diseases that are caused by rogue proteins that the immune system struggles to lock onto. The genetic code expansion methods allow the researchers to increase the alphabet of available amino acids encoded by DNA. And by coupling metabolic engineering techniques with genetic code expansion, the researchers were able to create a system that produces nitrated proteins autonomously. And because of the nitro-functional group chemistry, the amino acid that they picked as their target for the project was unconventional, and many scientists with their field may not have expected that it could be made using biosynthesis. The next step for this research is to optimize their methods to synthesize higher amounts of nitrated proteins and expand this work into other microorganisms. The long-term goal is to further refine this platform for applications related to vaccines or immunotherapies. I think the implications are interesting, and that you can take a bacterium's central metabolism, its ability to produce different compounds, and with a few modifications, you are able to expand its chemical repertoire. The nitro functionality is rare in biology, and absent from the standard 20 amino acids, but they showed bacterial metabolism is malleable enough that it can be rewired to create and integrate this functionality. As bacteria are potentially useful drug delivery vehicles, I think they have created a tool that have leveraged the ability of bacteria to produce target antigens within the body and exploit the ability of nitration to shine a light on those antigens at the same time. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.